Hi, I'm Greg. I've just recently finished my PhD here at Birmingham, so I'll be telling you a bit about that work today where I was exploring the human gut microbiota as a reservoir of antibiotic resistance genes. So originally my talk was meant to be showing combining long-read metagenomic sequencing with high-C data to link resistance genes to their hosts, but I've been working on the long-read stuff currently, and it took a bit more time to get the sequencing on, and the analysis is taking a while, so I won't be able to show you any of that today, unfortunately, but I'll be showing you high-C to link resistant genes to the host, just without the long-read stuff. I'll come a bit back to that at the end. So just as a bit of background, the human gut microbiota is home to a wealth of bacteria. Most of those have a commensal or mutualistic relationship with the host, which means they help digest food, they produce vitamins, they have immunoregulatory functions. But as well as those commensal bacteria, the gut can also harbour opportunistic pathogens. So these are species of bacteria that can live inside a healthy human without causing any problems, but given the opportunity, if someone becomes immunocompromised or vulnerable in some other way, they can cause infection. And the most famous ones and the main ones causing infection, I guess, are from the families Enterobacteriaceae, like E. coli and Klebsiella, and then also Enterococciae, like Enterococcus faecium and fecalis. And there's been a big rise in multi-drug-resistant infections caused by these opportunistic pathogens originating from the gut in recent years. And we know, as well as harbouring these opportunistic pathogens, the gut also contains many antibiotic resistance genes, collectively termed the human gut resistome. And because of how the gut microbiome is, with a densely packed bacterial cells and thick mucus layer lining the gut, it's a really great environment for horizontal gene transfer. So there's potential for a lot of horizontal gene transfer of resistance genes in the gut. And because of this huge rise in multi-drug resistant infections from opportunistic pathogens originating in the gut, there's a real need to characterize the human gut resistome and find out which bacteria are carrying the resistance genes and whether commensal bacteria can act as a reservoir for them and transfer them to opportunistic pathogens to allow this rise. And from metagenomic sequencing alone, you can see which resistance genes are present and which bacteria are present, but actually linking those data is quite difficult, particularly if the gene is on a mobile element like a plasmid. When you assemble it, it will be on a plasmid, and it's hard to then link that to which bacteria it was in a complex microbiome sample. So my PhD aimed to use chromosome conformation capture techniques, known as 3C, or a derivative called high-C, to link resistant genes to their hosts in the human gut. So that basically works by incubating your sample in formaldehyde, and that allows the DNA inside the cell to be cross-linked. So this could be distant regions of the chromosome cross-linking with other DNA that's in close proximity inside the cell. So that could be a distant region of the chromosome, or it could even be a bit of plasmid DNA being linked to chromosomal DNA. And then by lysing the cells and digesting the DNA, you're left with these fragments of cross-linked DNA that you can then ligate together. And when you remove the cross-link, you're left with one fragment of DNA where one half of it is one region of the chromosome and the other half is a distant region of the chromosome, or it could even be half chromosomal DNA and half plasmid DNA linked together. And then by sequencing that and analysing it, you can find these linked pieces of DNA, and you can know that they were from the same cell originally. So you could say this plasmid DNA is linked to this chromosomal DNA, and by identifying the taxonomic classification of that chromosomal DNA, it will tell you where the plasmid originated from. So I performed this technique on 4 different stool samples, and I used a spike-in of Acinetobacter pittii strain, it had resistant genes on both the chromosome and the plasmid, and that was just to sort of validate the bioinformatics as I did it. So I developed a bioinformatic workflow to use this data to link resistance genes. I've called it the H-large workflow, which is host linkage to antibiotic resistance genes. I'll just give you a brief overview of that now. So you have both a shotgun metagenomic library and also this high-C library. And the shotgun library is used for metagenomic assembly. And from the assembly, you can find contigs that contain resistance genes. And then you can also use that high-C data to help bin these contigs in assembly. And a bin is essentially where you group contigs that are deemed to be from the same genome, and those bins are then filtered. I then take my Hi-C reads and map them back to the assembly, and then I can find my cross-linked fragments of DNA by looking for intercontig reads. And they're read pairs where each read of the pair maps to a different contig. So that basically allows you to find the cross-linked DNA, because if you have this set of reads and you assemble it into longer contigs, if you take a normal fragment of DNA and you map the reads, this paired end reads back to the assembly, they'll often map to the same contig because they'll be quite close together within the genome. But if you took a cross-linked fragment of DNA, these will be distant regions of the chromosome that are likely to be on different contigs, or it could be on plasmid DNA and chromosomal DNA. So read 1 will map to one contig and read 2 will map to another. So that's what we're calling an intercontig read pair. And by finding those, we can find these cross-linked fragments of DNA. So those intercontig reads are filtered to try and remove any noise. I won't go into that detail today. And then I find links between resistance gene contigs and other contigs. And that's then filtered again to try and remove noise. But in the end, I find, I look at all these contigs that are linked to a resistance gene, and I look at which bin those linked contigs were placed in to identify the host of that resistance gene. So, as I said, I performed that on 4 human stool samples, and these are the results of that. I was able to link 87 resistance genes to contigs from their host. And first of all, just looking at these ones highlighted in purple here, these resistance genes, these were from the spike-in that I added, and you can see in all the samples they all link to Acinetobacter pittii correctly, including MPHE and MSRE, which are both carried on a plasmid. And then looking at the non-spike-in resistance genes, just the ones that were naturally in the sample, some of them were linking to multiple hosts, some of them single hosts. Just as a few examples, these B genes were present in all 4 samples, and they were often linked to multiple gut commensal species, particularly in H4, where it linked to 4 different gut commensals. And this gene is known to be quite promiscuous in the gut, so this could imply that this gene was transferring within these samples, and these were all in the same class in Clostridia. And then just another gene to highlight was CFRC, was in a couple of samples, and this is a quite clinically concerning multi-resistance gene in C. diff infections, and it's on the rise. And I managed to link it in 2 samples to 2 different gut commensals in the same class as C. diff. So potentially they might have the ability to transfer to the opportunistic pathogen C. diff. But you can see that the links were limited by the binning process. So a lot of these links down here were either in a discarded bin or unbinned. And that means I was able to link the resistance gene to a contig, but because that contig wasn't binned during the binning process, I was unable to actually taxonomically classify that using this workflow. So the binning needs to be improved to try and cluster the majority of contigs into bins to improve these results. So next we did some culturing to try and isolate some of these hosts of resistance genes to look more at the genomic context of the genes, but also to see if we could culture the same stuff that Hi-C was showing us. So to do that, I basically just grew up the stool samples in anaerobic broth with and without antibiotic, and then used qPCR to find genes that were enriched, and then did a load of colony PCRs to find isolates that had those resistance genes. And once I had those isolates, I then whole genome sequenced them. I did both long read and short read, and I did the short read sequencing with microbes NG. And then I did a hybrid assembly of those, and from that I was able to look at the genomes, find resistance genes, classify them, look at the context of the genes, but I was also able to compare it with the Hi-C data from these same samples. So looking at how they compare, I'll just show a couple of examples. Some of the culturing data was consistent with Hi-C. So this Colin Seller genome contained TET-W on the chromosome. And if we look at the Hi-C results from the sample that this originated from, we can see that TET-W was linked to Colin Seller. 11% of the links to this gene were to Colin-Seller. And when I take all the contigs that this gene linked to and map them to this isolate, 11% of them do map. So these links were to contigs that then align to this genome, which we know does contain TET-W, so it was correct. And same with this Bacteroides solocylaticus. It had 4 different genes, and they were linked with high C, and again, they did map back to the genome. But there was some examples where high C had failed to link the resistance genes, or at least failed to classify the hosts of those. So this Streptococcus parasanguinus had 5 different resistance genes, and from the high C data, you have no idea that these genes could be in Streptococcus parasanguinus. None of them ever linked to that, just in the high C data. But if I do take the linked contigs that these genes had been linked to, some of them do map to this genome, which we know does contain them. So high C had correctly, or H. large had correctly linked the contigs to, linked the genes to contigs that were originating from this genome correctly. But again, because those contigs were unbinned from the high C data alone, we weren't able to see that those resistance genes were in Streptococcus. So again, this highlights the need to improve the binning process. So in summary, 3C techniques can be used to link resistant genes to their hosts, but it is limited by the ability to bin and classify the contigs. And that's something I'm working on now in Josh Quick's group here in Birmingham. I'm trying to improve that using long-read sequencing to get better assemblies. And just some very preliminary results. I've finally got enough DNA to sequence, and I've been sequencing that on the PromethION using Oxford Nanopore technology. And I'm getting hundreds of gigabases of data for each sample that takes ages to assemble, but just from some very preliminary stuff, if you look at the assemblies of, just for this sample H2, the short read assembly versus the long read assembly, it's about the same length, but it has much better N50, as you'd expect, because you're getting longer contigs. And then just from some quick binning, we are getting a lot more bins. And particularly if you look at the proportion of contigs and the proportion of the length of the assembly that's clustered into bins, we're getting great improvement with the long read stuff. So we're getting over 70% now being clustered into bins, which is a big improvement. So hopefully this will allow me to link more genes to their host, because I'll be able to classify them more, but I haven't done that work just yet. I think it's important to note that you can never know with absolute certainty the host of resistance genes just from the high C data, and it's more of an indication of where they might be. And I think it's good to couple it with culturing, because you can then actually isolate the hosts and look at the genomic context and be certain that that's where the resistance genes are. But I did show that gut bacteria are an important reservoir of resistance genes. There was a lot of genes that were widespread in commensal species, particularly aminoglycoside and tetracycline resistance genes, and mainly in gram-positive species, so that should be looked into a bit more. So thank you very much for listening, and just thank you very much to my supervisors, Willem van Schaik and Sarah Kuhner, and thank you to Amanda Rossiter, because she lent us, or provided us with the stool samples from her stop colitis study. And then thank you to the rest of the Van Schaik lab and McNally lab for being with me throughout. And thank you to the Quick lab, who I'm currently working with doing the long read stuff. Thank you very much.